Okay, welcome to CULINOLOGY 101. Okay, so for this week we're going to talk about introduction to the cell. Okay, it's more towards looking at the components in the cell. Alright, in the cell is very important for you to understand is because those animal cells or the plant cells, okay, which is basically made up of your food ingredients, they contains the components that you should be aware of. Okay, let's look at the learning content. Okay, the first is able to differentiate between different type of classes. Okay, there are two classes we're going to introduce, the prokaryotes and the eukaryotes. Then we're going to look at the microscopic observation of the cells. Then we're going to look at the different type of the components. Okay, then we're talking about the components inside the cells like nucleus, ribosome, endomembrane systems, okay, and also the energy converting organelles. Okay, let's look at the first one. First, you need to differentiate between different type of cells. Okay, the one that you'll be using, right, okay, is most likely going to do with either one of these, okay, prokaryotes or eukaryotic cells. So what's the difference between these two types of cells? Prokaryote cells basically are the very simple type of cells, like the bacteria, which is the single cells. Okay, later we're going to talk more about what is really the difference here in terms of the cell components. Whereas the eukaryotic cell is like humans, okay, different type of animals, plants, and the upper level type of the fungi. Okay. So when you're looking at the difference between different cells, one major difference between the two classes is indicated by the names. Okay, if you're looking at the prokaryotic cells, pro means before, okay, karyon means nucleus. So it's basically a cell that is before the nucleus, a true nucleus. There's no, for the prokaryotic cells, they are DNA which is basically the genetic components, okay, that control the whole cells. They are not partitions. They are not basically they are not separated, okay, and they appear on the earth before the eukaryotic cells, yeah, which means the prokaryotic cell comes first, then only comes the eukaryotic cell, which is more complex. U means true, okay, karyons means nucleus, yeah, so they have a nucleus basically in the eukaryotic cell, the largest organelle with membranous envelope that contains the DNA. The DNA components is the genetic materials in the cells, very important, and is being separated away from other type of organelles, okay, for eukaryotes. Okay, let's look at the prokaryotic cell first. Okay, prokaryotic cells is the like bacteria that you encounter in the environment. Okay, we also use bacteria for certain type of food, like for example the yogurt made of yogurt, cheese, okay, fermentations, alright, like for example the what they call the fermentation of kimchi as well, we use bacteria. Okay, so all these are considered as prokaryotic cells, yeah. So using, as you know, bacteria got many different type of different types of bacteria actually, okay, and they can be differentiated based on these components here. Yeah? So usually if you're looking at prokaryotic cells, they have a simple flagella, okay, then usually they have a cell wall. Sometimes they have a sticky capsule as well to stick on their host or stick on certain type of surfaces. Then they have a plasma membrane. They have the cytoplasm, which is the interior component over here. Okay, the interior areas. Okay, it's called cytoplasms. Then they have a DNA. Basically, is the genetic materials that used to control the whole the cells. Yeah. What they don't have, right, is basically a nucleus. You can see that there's no nucleus. They only got a DNA suspended in the cytoplasm, yeah. And they don't really have a membrane-bound organelles as well, compared to eukaryotic cells, yeah. So, as I mentioned just now, that there are many different type of bacteria. Okay, these type of bacteria, right, we can actually differentiate based on their cells. Okay, the cell wall characteristic, their shape, whether they are the rod shape, okay, look at it, rod shape, they are the cocci shape, which is the circular shape, or sometimes they have a spiral shape. Okay, they can look at the different type of shape. in your microbiology, the subject, okay, later on you're going to learn more on this, yeah, okay, but now it's just introduction to the cells. Other than the shapes, right, there's a difference in terms of their cell wall or the outer membrane characteristic. Okay, how you differentiate is basically through staining, okay, which means that they stain on the surface. They're using different type of staining and they stain differently. For example, if it's gram positive, they actually can stain with a violet blue, and then they can appear as the purple color, yeah. This is because of they have this what we call the peptidoglycan cell wall. Because of this peptidoglycan cell wall, they can be stained by this violet blue. If you're looking at the gram-negative type of the bacteria, they actually stain with red stain, yeah, because of the lipid. Okay, because of the lipid at the outer membrane, that's why they can be stained with the red stain. Alright, if you try to stain with this one, it doesn't stain, okay. So because of the different type of the cell wall outer membrane characteristic. So other than shape, how they stain at the cell wall, some of them they also can be differentiated based on their flagella system. Some they have the single tail, multi-tail, some they have front and back, okay, some they have a multiple type of the flagella system, yeah. If you're looking at the eukaryotic cells, just like animal cells and human cells, okay, which is eukaryotic cells, is much more complex, okay. For the eukaryotic cells, they are much more complex in terms, you can see that they have more components in there. This one is the animal cells, and this one is the plant cells, yeah. Okay, they are structurally more complex. The complexity is actually reflected by their functionalities, yeah. Eukaryotic cells usually they have more functions. Because as you say, you can see that the humans, they are made of thousands of cells. They are all connected with each other, talking to each other. That's why they need to be more complex. And the chromosome between the nucleus, right, is being membrane, separated from there. Okay, there's a few reasons why they separated, yeah. The reason why they separated is so that they got more distinct, able to control what is in the inside the nucleus and what is outside. They can distinguish it. Okay, much more clear. Okay, much clearer separation is more, is better in terms of controlling how they express, how they move about in the cells, yeah. Okay, because of the complexity, they need to have more control towards it. And then some of them, they have a more complex flagellal system as well, for example, like sperm cells, okay. Other than that, usually they don't have this tail, yeah. If you're looking more closely into a cells, they have this what we call their organelles in there, okay. All these organelles, all of them have different type of functions. We're going to go through one by one later on, yeah. Okay, so what we have is, for example, they have a rough endoplasmic reticulums, they have the chromatins, they have nucleolus, they have mitochondria, they have smooth ER, okay. Then they got the nucleus, then they got Golgi complex and so forth, yeah. Much more complex. So you can also look at it, okay, as a summary. The prokaryotic cell is much more smaller, okay, and also much more simpler as well. Whereas for eukaryotic cells, they are much more bigger. You can see, yeah, this is very, very small, like bacterial cell, very small. This is the eukaryotic cell. One cell is so big, okay, and they are much more structural complex, and they have organelles in there, and they have a true nucleus, okay, that control the genetic materials. And the size as well, you can see, because of the size, these do not require a nucleus. They can move much more freely between the outside and the inside, okay. So if they want to transfer the cells from one end to the other end, much more easier. Whereas this one require more complex because of the bigger cells as well. They need more control towards their movement from one end to the other end of the cells, okay. The bigger, the more control needed, okay. Okay, how to observe these cells? You actually can use the microscope, yeah. You need the microscope to observe the cells, especially what is happening inside, okay. If you're looking at this actually shows that whatever is below here, our naked eye cannot see, and we require the aid of the microscope, yeah, to looking at the more detailed structure. The size of the cells, right? Why is it limit in that cells? The size is because it cannot be too big. It cannot be too big is because you remember that they need to actually transfer from one end to the other end the components. Okay, so they require a certain size. Okay, so that they can undergo effective chemical reactions, uptake of oxygens, okay, removal of waste, as well as the uptake of nutrients. Okay, cannot be too big as well. We have different type of the microscopes. We got light microscope, we got fluorescent microscope, we got electron microscopes. Okay, these different type of microscope actually is because of the resolutions. Okay, what it means by resolution is that to distinguish two objects. Like for example, you can see from here, this one is a low resolution, you can't even distinguish at all. Whereas this one, you can see that you start to be able to distinguish, and this one is much more clearer. Okay, different type of microscope got this ability. Alright, so some microscope you see that it's only one dot. Actually, there are 3 chromosomes in there. Whereas this you only can see one spot. So you require a higher end, like for example, electron microscope, to go from here able to see only one dot to able to see 3 chromosomes in there. Okay, so it's more towards the resolutions. Okay, in the lab, that for students usually we are using the light microscope. We can actually adjust based on the bright field or dark field, and that actually differentiate between the different type of the cells here. Usually you will require to stain the cells in order to look at more details, because different type of staining actually target different type of structure, and you can actually look at the different structure by differentiating the color you look under the microscope. Okay, bright field is where the light directly shine through your sample. Okay, this is the sample. Okay, directly look through your sample. Okay, this is a stage your sample usually put on here. So when you're looking at bright field over here, the light directly shine through your sample. That's why it appear bright. Whereas for dark field, you have what we call a dark field, the light stopper here. So what happens is that the light do not actually go through your sample. Instead, it will reflected out from the side. That's why you can look here with the darker background, but they have a different type of usage here. If the cells is illuminated, like the fluorescent, it can light up by itself. You can see that there are some light coming out from the cells. Okay, basically they use it for different shade lah. If you can't really look at the bright field, you can use it the dark field and try to see whether you can see more details or not. Okay, if you go through the fluorescent microscope, as I mentioned, you can do through staining. Staining is very good. It can differentiate different type of the components, like the proteins. If the cells contains a lot of proteins, you can differentiate the protein side compared to the lipid cells. You can see here, you can see these are exactly same cells, but then here you can see the red color maybe is to say that is the protein. Maybe here is looking at more towards the cellulose or something like that. Okay, different type of staining can differentiate different type of components here. Basically, you can gauge more of what is it composed of. Okay, the cell. If you're looking at the electron microscope, okay, it's a very big machine and very expensive as well. You can look at very detailed type of the structure here. But of course, to obtain this kind of the image is not simple here. The person who operate the electron microscope need to be very skillful. Okay. Okay, now the last section, which is looking more towards the eukaryotic cell components. Okay. So the first thing I want to introduce is the nucleus. Nucleus is very important for a cell because it contains the genetic libraries. The genetic library is basically the information to create the worker or the proteins. Okay, the genetic materials is where you use it to build the proteins, the enzymes, the different type of their protein-based components. And each of these protein components got a function. Okay, it can be a structural function, it can be a certain type of the enzymatic functions and so forth. Yeah, That's why if your genetic material has changed, what happened, right, is that the whatever they produce can be changed. Yeah. For example, some of the people, they cannot drink milk. Why? Because of their lack of a compost. Okay, like the milk, they cannot tolerate the lactose in the milk. Okay, the reason is because their gene, the gene has some sort of like the sequence that is not right. Okay, that they create a sort of like defective type of the lactase. Okay, an enzyme to digest the lactose, and so they cannot digest the lactose. The lactose went to the intestine, and the bacteria start to consume it, and they have certain type of activity over there that cause all the discomfort. Yeah. So you can see one more thing here. Most of the genetic material will be in the nucleus. Some they actually contains in the mitochondria and the chloroplast. Yeah, chloroplast in the plant, yeah, which is the photosynthesis of for the plants. Yeah. Okay. If you're looking at the structure of the nucleus, you got the nuclear envelope. Okay, nucleus inside. Okay, and if you're looking at this, they have a two membrane. They have an inner membrane and the outer membrane that actually separate these the genetic material out from the cytoplasm. Other organelles are outside there. A structure that divides the nucleus from the cytoplasm. Yeah, cytoplasm is outside, the nucleus is inside. Yeah, cytoplasm is the outside area. Yeah, not outside the cell. Like okay, like what we have here is like this is the cell. Okay, this is the nucleus. So this area is cytoplasm. Yeah, this is outside the cell already. Okay, so here they control what goes in and what goes out. Okay, so basically if you're looking at this, the genetic material over here, okay, the DNA basically will be transcribed, okay, transcribed to what we call the mRNA. Okay, the mRNA will be go through the membrane, okay, and being translated to protein by the what we call the ribosomes here. Okay, so they need to pass through this to be translated into a protein or active enzyme or structural protein or things like that. Okay, and the membrane also solutes and macromolecules from passing freely between the nucleus and the cytoplasm. This is very important, yeah, especially for large cell like eukaryotic cells, okay, to have more control towards the cell, yeah. So if you're looking at the outer membrane, okay, they have look at this. They have what we call a bit of a rough surface, yeah. Or the rough surface is due to this ribosome. Okay, started with this ribosome, and this ribosome is very important. Okay, so this ribosome is basically the components that actually translate the mRNA into a active protein. Yeah, protein, as I say, it can be a structural protein. Okay, means to build as a structure. Okay, it can be used structural protein. Then it can be an enzyme. Okay, it can be a lot of other things as well. And basically, this the membrane over here is continuous with what we call another organelle called the rough ER. Okay, rough endoplasmic reticulum, yeah, ER. Why is it called rough? Also, same thing is because of the presence of the ribosome at the ER. You can see over here, yeah. You can see over here, this area, the blue color. This is the ER, and you can see that there's a light blue color, that's ribosome here. So basically it continues over here. Why it need to go through all this is because as the okay, and the protein being formed by the translated from the ribosome and is being processed. So processed out is being packaged, like for example folding, it can be added with another components into the protein and so forth. Okay, all this so that they can create a protein that with a function. Okay, a functional protein, yeah. Okay, protein with a functions. It's like a car that is being manufactured, then they put in door, side mirror, and so forth. Yeah. Okay, the ribosome that I have mentioned again and again, right, is very important. Okay, is basically to translate. You can see from here that I draw over here DNA, mRNA, get out from there. As it go along here, the ribosome will translate the mRNA into protein. Yeah. So ribosome can be suspended or attached to the membrane, either the nuclear envelope membrane or the endoplasmic membrane, and it carries out what we call protein synthesis. Okay, to produce the proteins here. And the next one, right, is basically the endoplasmic reticulums. We have smooth ER and rough ER. The one that continuous with the nuclear membrane is basically the rough ER. Okay, after the rough ER, then is what we call smooth ER. Why is it called smooth? It's because the smooth ER doesn't have the ribosome. That's why you see the surface is smooth. Okay, there's a difference between these two. Yeah, for the rough ER that continuous from the cell membranes, okay, is basically the rough ER have functions where is a membrane factory for cells, adding membrane proteins and phospholipid to its own membrane, as well as use it to produce of secretory proteins. Okay, the proteins like, for example, the cells of the salivary glands, your saliva, okay, are rich in the rough ER to secrete an enzyme into the mouth. Okay, is to transport the proteins into the mouth to digest whatever components that you ingest. Yeah. Whereas for the smooth ER, okay, is lack of ribosome, so no protein synthesis. Okay, is the membrane is highly curved and tubular, forming interconnected system through the cytoplasm. Okay, it has a diversity of enzymes that build there. It doesn't have the ribosome, but it has enzyme built to the surface of the rough ER, the smooth ER. Okay, it has many functions. For example, it's used to synthesize steroid hormones. So as I mentioned already, as you go along here, you will be utilize some of these organelles to enhance, okay, the protein functions or to build certain proteins with a certain functions. Yeah. So once it go through that, the rough ER, smooth ER, then it will reach what we call the Golgi apparatus. So the Golgi apparatus is basically what we call a refinery, okay, or a warehouse or the shipping center for the proteins here. It finishes whatever proteins is stored and distribute the chemical products of the cells out, can be between the cell or out of the cell. Then we call the lysosome. Lysosome is another organelle. Okay, lysosome as the in Greek means breakdown body. Okay, it break things up. Lysosome structure is an enclosed sac. Okay, inside contains a lot of digestive enzyme. So in the lysosome, inside contains a lot of digestive enzyme, and the digestive enzyme is used to break down components that no longer needed, like for example protein, polysaccharide, fat, nucleic acid, they do not need. And after breaking down already, all these components, okay, the small component will be recycled to build a new components. Yeah. Okay, last but not least, okay, we have another very important organelles in the cell, which is the energy converting organelles. Yeah, the organelles we mentioned here, very important to have this energy converter because it's used to power the cell. Okay, without this, your cell doesn't have energy. You cannot use it to provide energy to work. So there are two types of cellular power station, chloroplast and mitochondria. For plants, you contains these two, chloroplast and mitochondria. For animal cells, it only contains the mitochondria. Yeah. Okay, let's look at the first one, which is the chloroplast, which is plant-based. So what is chloroplast? Is basically is an organelle in the plant that use it to produce photosynthesis. So with the chloroplast in the cell, the plant cells, it can absorb, okay, or convert the sunlight into energy, okay, or a process what we call photosynthesis. Yeah, conversion of the light energy from the sun to a chemical energy of sugar and also other organic molecules that can be used by the plant cell. Okay, so you can see from here, this is the leaves. Okay, so you can see there are a lot of chloroplasts within the leaf cells over here. Okay, the chloroplast is represented by the green component over here, okay, and use it to absorb the sunlight and convert into the energy, chemical energy. You can see from here, yeah. So this is a leaf, this is the plant cell, and inside the plant cell contains a lot of the chloroplast, yeah. So same things here, inside the plant cell also contains the nucleus and other organelles, yeah. So for mitochondria, it will in both plant and animals, okay. For plants, in the morning when there's sunlight, they will use chloroplast to produce the energy components, but at night when there's no sun, they use the mitochondria for the respiration, yeah. For animals, they only contains the mitochondria for respirations, yeah, to convert into the energy. So what is mitochondria? It's a site of cellular respirations, okay. It harvests the energy from sugar and other food molecules and convert it to another form of chemical called ATP, okay. For the energy, right, it can come from a lot of components. You will learn more from the food biochemistry. Okay, I'm not going to talk more about this, but ultimately, all these components from the food, okay, especially the carbohydrate, will be converted to sugar. The sugar will enter into the mitochondria and converted to a chemical energy called the ATP. So this is the structural component of mitochondria. You can see that they have a lot of the units over here at each of these. You can see the reason why you have this internal intertwine over here, right, is because to create the surface area, yeah. Okay, and each of these is an area where they convert the sugar molecules to the energy components here, okay, called the ATP. We're going to talk more about this one, especially when we talk about the conversion from meat, from muscle to meat, okay, because it involves this as well. Basically, you will see that the glucose, okay, this one is very relevant to the later chapter here, the conversion from muscle to meat. Basically, the food they contains rich in carbohydrate. The carbohydrate will break down ultimately to the glucose, okay, and you'll be converted to what we call the pyruvate, okay, glucose to pyruvate in the cytoplasm. Okay, this happens in the cytoplasm of the cell. Will go into the mitochondria, which is this. Okay, this one will happen in the mitochondria. The pyruvate will go through the Krebs cycle, and then the components, okay, what we call the oxaloacetate. The oxaloacetates will go into the oxidative phosphorylations, and you will generate the ATP. Yeah? Okay, so this is the whole what we call the biochemical pathways. Okay, it's more complex than this. Yeah, this one is just a summary of the steps. Yeah, okay. I will go through this more in the conversion from the muscle to meat. Okay, don't worry about this, but at least you know that the mitochondria got to do with the conversion from muscle to meat, yeah, and it has a very important impact to the quality of the meat as well, that I'm going to explain later on in that chapter. Okay, so that's it for this chapter, okay, introduction to the cells. Okay, so if you have questions, please ask, okay, in the Padlets. If you do have no question, please indicate no questions in the Padlets. Okay, so thank you very much.